Hello, everyone. Hello for this seminar. Whoever is interested, how life started, I want to give you an overview on lab experiments where we try to explore how RNA, early Earth, comes together. Here we go. Early life is quite old compared to space Big Bang, 14 billion years ago, about 4 billion years ago, where life likely started. But the connections are not obvious. We have to go for biology, look how evolution backwards makes Darwin evolution. But then the only life we know is the life started on early Earth, and how that's connected to the astrophysics needs to be still resolved. So if you think about yourself, what are steps along the logical timeline? I'll give you my perspective, where early Earth gives us rocks, porous, interesting things. Cells are then the latest best development of life. Life could go everywhere, but I would argue it started in some rocky environments. Proteins have been important to do that because proteins are able to run the metabolic networks, run the channels in these first cells, all the complication there. But to have proteins, you have to have a ribosome, you have to have a genetic code, you have to have tRNA, you have to have RNA made out of nucleotides. Additional molecules will be necessary for that too, but the core of it will be RNA evolution. And the question will be, where do vesicles and cells start? My argument will be that that's actually late development. You don't have the vesicles early on. You actually don't need them because the rock is providing those environments there. Metabolic networks is hard to imagine early on with very simple molecules if you don't have the proteins guiding the reactions. And also proteins, to start with proteins, the first step, you lack that encoding capability to make your proteins in a very specific sequence. So this is the timeline, and you can always ask, this is a scientific question, because you might say, we never know how these things have evolved. We think we can learn, I think we can learn a lot because we can test hypotheses. We can have a look. Is this how life could have made it? Let's go for the lab, do it. Does it work? Doesn't it work? And by that, we can figure out how life could have started there. And partially of that is that we focus on these rocks, especially our water interfaces have, I'll focus today, have interesting capabilities of accumulating the molecules at the interface, replicating them. Molecules, I mean RNA, which replicate the sequence of the RNA. And for example, if you add lipids to that, they actually form vesicles where inside you have more of the RNA molecules than the outside. So we think it's a dual-fold thing. One hand side of physics, which is concentrating the molecules, making the large molecules better concentrated. It's a self-amplification of size, and the sequence which is replicating, amplifying the sequences from one molecule to the next, and therefore amplifies the sequence on it. Again, as we have life and you have complexity of replication, these cellular entities where a lot of proteins are at work, minimal cells, probably more 1,000 than 500 proteins, a lot of bases to make that work, and very complex coordination of cell division, replication. If you look for the replication, which you need every time you divide the cell, this is the machinery. Already that machinery needs a lot of proteins, which is splitting the DNA, replicating the DNA, and make it work. So that's too complicated for early life to do it. And it would be difficult to think about how to make that. So we think you rather think about short pieces of RNA, which can assemble other short pieces of RNA, which are shorter, but half the size, and then link them together. We'll focus on that. Strand separate, have a non-equilibrium system which pulls these molecules into lower salt regimes where the strands can separate back again and the process repeats, and each of that cycle gives you twice the amount of the informational molecule you have in the beginning. To feed that, you have to think about how to make those shorter strands, and one starting molecule we favor, or the experiments favor it. In our hands, is 2,3 cyclic nucleotides. So you have this special cyclic dehydrated form of the phosphate back there. And if you have that in a dry state, and we operate here at quite alkaline conditions, these are polymerizing on the backbone, and you get the RNA pieces. Please write down your questions you have, because we'll have 15 minutes question session after the talk. So it's always best if you write down your questions. So what this gives you is a way to replicate by mirror symmetry of one sequence is the template for the other sequence and replicates the other. What emerges from that, and we studied that with random sequences and a protein system to make it fast, is a network of sequences which are mutually replicating. Actually, the random sequence is immediately reduced to small amount of sequences, and that is developing, that's the argument, Darwinian evolution in these networks of replicating systems. We would argue that's the origin of life. So what then is the next step? Eventually, that you combine that with amino acids, make it into a ribosome, get tRNA going, and make that a first machinery to make then complexity increased of your evolutionary system. So we go through some assumptions which people use and think about the system and give it a bit of critical test on the experimental side. One of them, life emerged in the liquid state. Well, the early life was quite hampered by the moon forming impact with a very complex initial condition, which probably was special for the Earth, where you likely had in the beginning reducing conditions and quite a number of head start molecules to feed from. But eventually you have to survive a water world where dilution is imminent and you have molecules rather getting everywhere. And that, of course, makes things hard if you need to have a drying reaction, as you will need it for amino acids and for those ligation and polymerization reactions. So dilution is the big thing you have to be afraid of, and therefore going for a dry state is an important step. Nucleotide synthesis pervasively needs dry steps. And therefore, we think we have to have some dry environment where you can make these complex molecules, which are more complex than what you find on asteroids, and likely is important because those are the molecules which are capable to run these networks by replicating themselves, their self sequences. So what these volcanoes give us is porous rock. So in these pores, molecules can hide away from UV, can hide away and concentrate up. And that's the setting we would be very interested. Please note that if the CO2 in the beginning of the Earth, due to the faint sun effect, is not so warm, you could actually and should also think about ice conditions. So just open up a bit the possibilities that chemistry is not only running in the liquid state. With that, you likely would have a hard start for the origin of life. The second generally accepted starting point is that life is at pH 7, where modern life thrives. The interesting finding is if you go and leach ions at higher temperature out of rocks, you're not getting pH 7. And if you go to Iceland, Villa Vedlia here, you will measure a pH 9, 9.5, almost 10, which actually gives a nice warm water pool, but which you should not be afraid of. People argue that these pH, you will actually destroy RNA. That is actually not the case. RNA is pretty good in surviving high pH if we don't have divalent ions, which you anyway don't have at high pH because they're not very soluble. And if you restrict yourself not to jump for too high temperatures, it is actually thinking about that possibility. And we will see that also double-stranded RNA is possible at pHs higher than pH 10, which comes to a surprise and also surprise us in the experiments. So high pH is the possibility these volcanic islands are providing, and let's delve into what can be done at that pH. The other idea people are pushing in the field is that they think RNA can only replicate itself by ribozymes, complex folded RNA structures, which are very complex and long sequences, 50 mers, 100 mers, that they are driving the replication of the RNA. I mean, sure, at some point they will do the ribosome, as you saw, but the idea is actually in the approach we take here that this is not the core functionality of the RNA being catalytic. The core functionality is actually that the complementary sequence brings the ends of two strands together if the bases are complementary. So the catalytic function of RNA is its double-strandedness and nothing more. So what can be seen there is that this templated ligation, and I have here a nice magnetic model of that, where you do have your white strand, which goes through, and you have bound now, on the other hand side, two short pieces, which are complementary. Those are bringing, by the stacking forces, these ends of the yellow sequences together. And if they have this 2,3' cyclic phosphate I told you before, they are actually ligating. You need a pH 10, you need moderate temperature, and you get a ligation in a week of about 35%. That's a published result, CHECS 2024. We now have increased this yield about, and the kinetics of it, sixfold. So we get 50% yield almost in a day. That's possible if we go a little bit higher pH, which, for example, amino acids give us a good buffering system, but that's a longer discussion how to set the pH up there. What this means is that if we ligate here, for example, with one reaction, 8 mers into a 16 mer, we are replicating 16 bases in one reaction, so that about 50% yield gives us a yield per base of replication of 96%, which is very much comparable to what large ribozyme systems can do, and therefore it should be quite competitive for complex RNA structures. So there's a longer discussion which pH is good for the ligation rate. Higher pH is better, but the hydrolysis of the 2,3' at some pH and some temperature will take over, and then if really much temperature, higher temperature and much higher pH, eventually also the backbone cleavage will set in. So there's some sweet spot of this reaction, which seems to be about 10.4, and rather 15 degrees, 10 degrees, where the 2,3' is there for a longer time, and you get good ligation. Please note, without template, you get no ligation. You can also, because the ligation gives you about 50% of 5'2' link, which is not the natural biological one. It shouldn't worry us too much at the early stage, because if you have your template, the long strand where here the blue one is binding to it, you are getting the binding of the gray one, even if that link here is a 2,5' linked one. So even if you're a bit messy about that linkage, this is robust mechanism. A DNA template does not replicate. Could be because it's a B form, not A form of the RNA strand. And if your phosphate is at the end of the one strand, not cyclic, but linear, also nothing happens. So that's the expectation, but that will be the first catalytic function for RNA. It turns out this is a pretty precise way of replicating. If we change the sequences at the ligation site, only very strange combinations of two wrong mismatches is giving us a wrong ligation, significantly one. Otherwise, it's very much suppressed. We confirm that now with sequencing data. Shofia Megiezi was looking into this, where actually these are the 4 bases downstream here, which we probe by sequencing, and you get about 90–95% fidelity on your base, as you would expect from this reaction here. So it's actually quite precise. We can think of that this replication reaction goes on for a long time. The question we have not yet managed to fully solve is that we'd love to get this exponential, of course. So after the first ligation, you'd love to have some non-equilibrium system, or some waiting time or whatever, which separate the strands. New strands come onto both of those strands, either the gray one or the blue one, and replicate them again and again and again. So we are looking into that. We understand it much more, much better, and it's been our specialty in the past with PCR reactions, non-equilibrium sittings. So we think we will solve it, but give us more time. In theory, if you want to have a preview, what to expect, if you put it on a computer and you have about 100-fold excess of the short pieces, which is expected from an exponential polymerization dynamics, this is what you would get. You would get about a factor of 10 or more in replication over the course of a week, if this is at the sweet spot, given our measured kinetics. That's the ultimate experiment we hope to be able to do, because then we could run this with random initial sequences and have a look how the system actually evolves, which sequences are faster, are slower, can we replicate with a high fidelity, keep the sequence, and run Darwinian evolution. So watch this space. We are optimizing this and hope to get this going better sooner than later. The idea of the setting is that you can replicate a long strand, blue here, by having all those short strands bind there and being actually independent on length when it comes to the time to replicate this, and should be actually quite efficient way to make longer and longer sequences. We've shown already sequences length of 80 or 90 might be generated from these short pieces by stitching them together. So in the field, people start with a triphosphate because they think they should mimic the biological activated molecules. As you've seen, that's not what we're doing. We think this constrained 2,3' cyclic phosphate is a better starting material. It's the outcome if RNA hydrolyzes the backbone, if it cleaves, not hydrolyzes, if it cleaves the backbone, you get the cyclic 2,3', and that automatically is a very simple way of recycling the molecule. If this 3' phosphate, triphosphate here gets diphosphate, you need to have quite a complex machinery to make it again. If you look for biology, that's one of the most complex proteins nowadays you can have. The idea in our point of view is rather that you have this 2,3 cyclic, and then later biology took over on the other side of the molecule, the activation. Now, what people argue is that you have this magically generated activated molecule from wherever, sigma or IVT proteins, and that will drive the system. So just activated RNA, that's the setting of RNA world, is the way you run the system, and that will overcome all those degradation pathways I've shown you here, degradation, and that will all fuse things together. Of course, you don't answer the question where these activated molecules come from, that non-equilibrium, but I would argue also that you need some other help from non-equilibria to put the molecules together, concentrate them, polymerize them, and also activate them to drive the system. And to give you some examples, I probably don't make friends here by stating that I'm not so much convinced about coacervates doing this, because the up concentration you see visually by fluorescence microscopy is not a reactive activation. So the theoreticians on coacervates will tell you that the chemical potential, which is driving the reaction, is actually the same inside and outside. So you bind to other molecules and they are locally highly concentrated, but it doesn't make you more reactive in this setting because it's an equilibrium system. Likewise, if you put things into a vesicle, it's hard to understand how the inside of the vesicle should be highly concentrated than the outside. And experiments which actually compared with vesicles and without vesicles under the same condition, the reaction without vesicles was more effective, was replicating better, polymerizing better. So also there, it's an equilibrium system where you insulate your system, and unless you have directed flow, it's not better if you do that. So because inside and outside is the same, right? So we think geological non-equilibrium give you actually real advantage in your reaction pathways. And I'll show you a bit a very simple way to think about these systems, what these are look like. So for example, you have a pore in these rocks and have nucleotides together. So the most simple way to accumulate them is you manage to enforce a directed motion across the pore. So that would be the velocity, given here in units number of molecules per second at area, given by the concentration C and the velocity V. So that can be evaporation, that can be thermophoresis. Those are the two typical ways we can think about it. What counteracts this flow is at the bottom of the chamber that diffusion will bring the molecules back into it. Okay, I mean, that's the only, you accumulate it like the molecules in the atmosphere. Basically, gravity brings them down, diffusion brings them up again. Here we think about RNA molecules, smaller chambers, millimeter sized, and that would, for example, give you an up concentration of a factor 10 for a given length of your chamber, given time you wait for these molecules to fuse back into the solvent. Now, the interesting thing is the following. If this molecule has the capability to polymerize in these settings by some dry cycle, for example, at the bottom, by some air bubbles, the diffusion coefficient will go down. And this exponential distribution here, length of the chamber, velocity of the accumulation, diffusion coefficient, is actually better accumulating the molecules. So you get a self-amplifying cycle, whereas the molecules get larger and larger, you accumulate it better because the better accumulation, it will make it easier to make it larger and larger because the concentration is not just given by the chemistry, but also by the physics of this co-accumulation as you go with it. The idea, and shown experimentally, you get these higher accumulations, more complexified molecules by these chambers if they can polymerize, and if, and that's a crucial thing here for RNA, it can bind to itself and actually make itself even longer. So that's the idea, what these pores can provide for the origin of life. It's an amplification of more complex molecules if they have the right capabilities. And the argument would be that RNA has that capability, and that gives you that exponential concentration amplifier in the setting, continuously allowing for nucleotides to come in. If they're used up, they diffuse out again. So you can feed the system from the outside continuously and remove the waste because it's an open system. You're not closing things out, and it's a non-equilibrium. So you get reactions going, which in equilibrium will not go, and here, because you drive it, could go. So that would be our prebiotic cell, and I'll show you examples of experiments on that. Can be driven by a thermal gradient, by evaporating water on the warm side, recondensing on the cold side. Can be a thermophoretic flow where you don't have an interface and don't need it because molecules tend to go from the warm to the cold side and therefore similarly accumulate at the bottom. But it could also be a completely open system where just wind blows over your rock sample and evaporation will do that. And if a flow here is triggering some convection, you can actually also have up here differential salt concentrations and run the strand separation problem, at least for a PCR reaction, it's been shown. So give you some idea how this looks like. If you look for fluorescence and prepare these systems, you actually see initially that you have a little bit more of a longer strand at the interface seen by fluorescence. Could also be an artifact of the fluorescence microscopy. But if you then switch on here the temperature gradient, which actually brings in the molecules, they can't evaporate, so they're stuck there. And you see that as a bright band, which fit to that exponential distribution I mentioned before. And you can do a full theory of that. And within minutes, you get more than 100-fold accumulation of more complex RNA, DNA molecules, the same for both species, more or less. So what does that give you? I give one example here where we have a molecule which can bind to itself. So that's a sequence. It makes hairpins, but if they are close proximity, the hairpins open up and they actually complexify and make a longer molecule. which in the solution is not happening because it's having these hairpin closed, but if they're higher concentrated at the interface, they actually make a big network. And that's what you see here already at the interface. So please note that you have a front side, which is cold, backside warm. They accumulate at the interface, and you actually see that accumulated part only if the water bubbles on the cold side jump back, the interface oscillates, and you actually see how this large beast of RNA molecules moves around. It's fluorescently labeled, that's how you can see it. And that's just, again, showing as it complexifies, it accumulates better and therefore makes rather complex structures, which normally without these gradients will never have happened at the interface. So in that sense, we would argue that we don't need to have cells where you need a big protein machinery. These non-equilibrium systems will bring things together. So you can actually have now, and that's the interesting part, in these settings where you have a gas bubble and a temperature difference, put in all the modern complex biochemistry. It's called a pure system, a translation system. So it makes from DNA, RNA, and then a protein. You can do that under a setting where the concentrations are lower and you basically kill the system. It's dead, it can't make proteins at that concentration. But if you have that flow accumulation, it actually complexifies itself and it shows GFP expression at the interface. Here's an air bubble in a temperature difference, and you see the accumulation of the GFP, and it expresses itself. The point we want to make is that the same setting, which potentially replicates RNA at the beginning, is still the same capability to run complex biochemistry. So evolution could run all the time at these interfaces and evolve complex life. You don't have to jump for cells in the first place. And that's been shown here in more complex, more different setting without these bubbles. So that's not yet published. The thing is published in Nature Physics, where the temperature gradient is giving you a cell environment. And there's a lot of discussion on how that complexified system is helping making that system go. So you see the expression only if you apply temperature difference in this setting. Now, what we were found very interesting to see is that where you have the accumulation of the RNA, you can also think if you put in lipids, those lipids should also do that complexification, right? I mean, they accumulate, make more complex small vesicles and then even larger vesicles, which means the diffusion coefficient goes down, they accumulate better, so you should be able to make it. And the combination of both should give you an environment where the RNA accumulates and you wrap it up into vesicles to make, in the long term, an environment which creates cells. And then the biological system of that complex biochemistry can actually self-encapsulating itself into vesicles, and that works very well. Those are not images of the lipid. What you see here are images of the molecules which are entrapped inside. So if you see these bubbles, it's not because you see the outside of the bubble, you see the bubble because you have a higher concentration inside than outside. So we find that quite fascinating that the same settings, which is not only helping RNA to replicate, it's also making the transcription translation system, it can also make vesicles. So we think it's a continuous bridge from these rocky systems towards cellular systems with these temperature differences. Potentially also evaporation should be good enough to do this. And it looks like a living system, but it's only driven by temperature. If you switch off temperature, it's all gone. So we think there's a missing link towards molecules. So bringing down the point again that these systems can accumulate molecules, that's what Christoph Must studied in the lab, thinking about many of these chambers, combining them, and have a look which of the amino acids are best accumulate the system and which of the bases are best accumulate the system. What we find here is that more hydrophobic nucleotides, valine, accumulate the best. And we actually also find that these ribozymes, which are activated, the 2,3' cyclic, also accumulate the best in this setting, which will get into a minute are actually the perfect conditions where the polymerization is best for RNA. So the idea is that these systems can accumulate even, hopefully in a big mixture of molecules, the right settings, and drive RNA evolution. And we studied that a long time ago, that if these molecules can polymerize, they would accumulate a lot better. Its title of the paper is this escalation of accumulation if you combine that. So you get an exponential increase of concentration if the molecules are capable of polymerizing. And they will actually dominate the cell, and all the other molecules who can't polymerize will be on the sidelines. Just want to mention that, yes, we use quite steep temperature gradients. You can make it even 100 degrees for 1 meter, but you have to wait for a year. Now, for early life, that might be okay. For our lab experiments, it's not. It might even be that evolution is getting the tighter temperature gradients to get by. If you're interested in that, that's the paper you want to check out. So the idea that RNA and amino acids cooperate is, of course, important because the tRNA, the ribosome, is making the proteins. But how does this system come together in the first place? We studied the polymerization of these 2,3 cyclic nucleotides. A, U, C is not polymerizing very much. G can polymerize a bit better, likely because of pK of the end groups, because that allows for acid-base catalysis of the reaction. U is also showing a small peak here. The funny thing is that you would love to go for high pH, but the high pH, you hydrolyze the 2,3 prime. It will activate the 5 prime OH to make the binding. The interesting thing here was now the following: if we add amino acids, and especially if we add hydrophobic amino acids, which accumulate best in our thermal traps, you get a huge enhancement of the yield of your polymerization. And there's a calculation from Judith Sponer indicating how the acid-base catalysis, general acid-base catalysis, can be thought about. This is nothing new because the hairpin ribozyme exactly works along these lines, but reverse in time. The idea is that the dry state is accumulating these lysine molecules, protecting the phosphate, removing a bit the water because of hydrophobicity, and have at the pK of the amine group the peak of activity where it shuttles through a proton through the reaction. This helps a lot in polymerization. You see here with valine how much more and more random sequences you get. And many other amino acids are helping too. It's a bit the trend that the hydrophobic help most. And this will give you a better way to get RNA polymerization. Here to the point that you get 5 mers of oligonucleotides with almost a random distribution of the sequences, as much as we can judge from the mass spec where this is based on. So amino acids help actually to make those 2,3 prime starting materials, which we need for the ligation. And there would be a longer story, and we're just preparing this, how chirality can be amplified by the polymerization and the ligation. The idea would be, and we probe this by an isotope-labeled RNA system, that it tends to have a homochiral polymer output. So the idea is that either you have a chemistry which only makes one-sided nucleotide monomers, which I think is an extremely hard question to get by because the amplifying system you need for that is just not existing for these small molecules. Amplifying systems are, however, possible for RNA by the replication of templated ligation. So we think that's a very good selection pressure for getting homochiral L or D. But we see that, interestingly, at the beginning of the polymerization already, where, for example, a G polymerization, if we have the same amount of D and L nucleotides, shows us an experimental result where the heterochiral DDL mixtures of that poly G is less pronounced as expected from a polymerization model than the homochiral ones. If you have a close look, the dry state for that polymerization actually means that the DD link is about twice more probable than the DL or LD link. And that's still also the case for the other bases, not as much. And that's a good head start to develop actually a mirror-symmetric chirality structure where the evolution of RNA on the right-hand side, right-handed and the left-handed would all work in the same mixture all the time, and only later feedback loops, biological evolution of stochastic sequences might have selected or will have selected one of the sides. So we can argue that if that decision is being done, it will be a stable setting. So if things are actually asymmetric between D and L, which is hard to argue chemically, but if there's some biological small bias, it will immediately amplify itself out and go by spontaneous symmetry breaking into either the directions. Just as an idea, if you think about origin of chirality, we think it's not the chirality of the single molecules, which is crucial, but the polymers, how homochiral they are. Happy to discuss that. It's here only in a sketchy way to show it. I want to end with systems which are combining now the replication and that accumulation. So the sequence amplification and the molecular accumulation. And we could only do that so far in PCR. I showed you how RNA in the future hopefully is able to do this, where we here have a polymerase, which is replicating a DNA strand. And we put that into a system where we have a temperature gradient. We mimic these pore spaces, of course, with a complex experimental setup where we have these well-defined temperature differences. In this case, we have a CO2 atmosphere, which gives us the advantage that as the molecules accumulate, the water of your buffer solution is evaporating as pure water and recondenses as droplets over here, of course, at much lower pH because the 1 bar of CO2 you assume here. Now those bubbles are falling down and have a twofold effect. The low salt will start to separate the strands and help it. And actually the pH might, on this system, in addition, also show the strand separation. So the idea is that you have short spikes of low salt because the surface tension of these water bubbles come down on your system. That can be shown here by Fred and pH measurements. If we flush CO2, which is now done, you see that the Fred shows single-stranded strands and the pH moved to 4 to 3 with the CO2. Would be interesting thing to have. You know, the CO2 helps us giving a high temperature Earth, but it could also help making the RNA separate the strands. DNA separate the strands in this case. RNA is the same. You charge up the C base and therefore separate the strands. So what is going on in the system now? We have these replicating molecules, but they can only replicate at the interface because only there the strands separate. And you do this replication with a polymerase, with a protein. So we need to do a trick at this point, but you see how strongly accumulated these replicated, very long DNA evolves. And that's the funny part of the system, that you actually get very long strands in this evolutionary pathway. So if we put in, and that's the experiment where we started, a 35 mer and a 70 mer, because we thought, let's have a look that the 70 mer can replicate better than the short 35 mer, because in a PCR reaction, we could show the short one is actually dominating because it can replicate a lot faster than the long one. All we wanted to show in the experiment that the long one, because of that accumulation, it will accumulate better and replicate better. That was our expectation. But anytime you do actually a Darwinian evolution experiment, it actually goes in directions not expected. And also this goes in that direction. We immediately saw quite long strands because the selection pressure apparently was so strong that only the long molecules could replicate efficiently at the interface by strand separation. We get actually concatomers of your initial sequence, of our initial sequence, and get sequence of 200 mers, 250 mers, which then face the problem that based on our analysis of the chamber, they have a hard time to separate the strands. So 250 mers are just at the limit of what the chamber can do if the GC and AT ratio is the same. Now, you might know, because of the two Watson-Crick pairs of the AT for DNA, AU, and RNA, base pairing, if you add more AT than GC, you can open strands much easier. And funny enough, this is what the sequences did. They were forced so much to be at the interface, only being there if they're really long. They actually threw away their sequence and only went for that selection pressure to make it long, and we get up to 1,000 mers of DNA in the setting. So we evolved from tiny RNAs, DNA, into very long DNA sequences. So that gives us hope that these non-equilibrium systems really drive evolution towards complexity. And that's done here with 6 hours. If we estimate for the ligation reaction for RNA, we would expect that in 30 days, 60 days. So we think it's experimentally viable to do that. And what we've seen on these sequences that we see actually a periodicity of 3 bases, likely because then they can have more ways to bind to each other, which is of course interesting because it might guide the way towards a genetic code with 3 bases in this setting. So these are the kind of dogmas, ideas we try to challenge by experiments. So we try to learn beyond the state of the art. And I have gone through those. And I will stop here a bit. I'll give you some minutes on, we can do that without temperature gradients. The replication, that's a eLife paper by Philip Schwintek, where the salt difference only is driving it. We actually even don't need CO2. And then longer discussion on the activation, reactivation. We try to get from the linear phosphate back to the cyclic phosphates. And these networks are, I think, that's what we expect for RNA. And we can look into the details of the DNA case for that. But I'll just jump over those. And also amplification. So we think these replicating networks with ligation offers many amplifying capabilities here to the point that they actually take over what's been discussed for hypercycles in a much more interesting way. That's experiment Toyabe is now professor in Japan. Check out his recent work on actually ligating and enhancing the fidelity in the ligation chain reaction. It's very interesting. So the hypothesis we are following, we think the polymerizing molecules accumulate exponentially better in geological pores. We first polymerize from 2,3 cyclic nucleobases at high pH. Seems to be the most simple way to do that with RNA, which then at the same pH do templated ligation, which will create replication networks. Chirality will be sorted out by having both chiralities in the same chamber. That's the idea. Experiments to be showing that, but also theoretical models confirm that. RNA replicates by those ligation chain reactions will make networks of sequences which co-replicate them. They typically tend to be 2 clusters, AT, A poor, T poor, because one replicates the other and doesn't make so much hairpins. So we think these accumulating molecular traps are very good, still can survive can host modern biochemistry and give rise to cellular life by creating vesicles in the same setting. So 2 shout outs for networking. Number 1, if you're near Munich, we have this network on website is molecularevolution.de. It's a CRC, it's a DFG-funded collaborative research center. We hope this is running for some more years, preparing their first extension. We have Molecular Origins Munich coming up. If you're in Munich, June 11, 12, you can still register, still put up your paper, posters, come by. It doesn't cost anything. It will be a big party on origin of life research for 2 days. Please stop by. And big thanks to the group. Some people have left already by now, and the funding from the Synergy Grant, CRC, and all of that can also lead to biotechnology. You want to check out Nanotemper, we actually use these temperature gradients to great benefit of pharmaceutical companies. And I'll end here by showing again the hypothesis and ask for questions. Thanks.
Thank you very much, Dieter. I don't know how you've do it. You've summarized something like 10, 20 years of work in a few minutes. Incredible, as usual. There's one aspect that I've been talking about for a long time that nobody in the prebiotic community seems to take seriously, and that's the fact that silica gel was ubiquitous on the early Earth at the rock-water interface. Silica gel is porous. You can have all kinds of gradients going through it. But nobody seems to be adopting silica gel as a support. Avinash Das, in a paper in 2018, did some polymerization experiments on silica gel, showing that you don't really need drying, but this kind of polymerization on the walls of the silica gel vesicles can be very useful. The other thing, that's a comment. The question I have is, you're hypothesizing, or part of your hypothesis relies possibly on evaporation, on drying conditions. But the surface of the early Earth was bathed in strong UV radiation. What is your opinion about the importance of UV radiation, either on the beneficial side, a la Adi Prof and Robert Pascal, or on the negative side? But thank you very much again.
Yes, I think silicate might be very good in buffering. We're here with Judith Bohmer right now. We discuss a lot of buffering systems. The pores might be a bit too small for the effects I want to show here, so it might add, and we added many things in the past. Some things don't help. We probably have to give another try for the silicate. In terms of UV, that's one reason why we go for these pores, because they will protect you. And I'm a bit agnostic when it comes to UV. We have a paper showing that UV likely played a role in the evolution of the genetic code. And RNA is very UV stable, but that doesn't mean that means that RNA could survive some UV, and it was exposed to it and therefore selected for it. But was it really necessary for the chemistry of making them? That's not. As you see here, we just assume we have nucleotides and start from there. And then if it's too much exposed to UV, you won't get these systems running. That doesn't work. Yeah. So
thank you very much.
Try to stay away a bit. Yeah.
Okay, thanks.
Yes, it is much more a comment, because this hypothesis is not very new, because in 2004, Martin and Russell have also said that pores are very important and essential because also of these kind of chain reactions of molecules and arrangements, even in interaction with the rocky surfaces. And with that, I think the origins of life, you are just consolidating this kind of hypothesis, which is since years, about 20 years ago also, is still, in the room. I would really say this hypothesis, you are just consolidating other generations which worked before on this topic.
I'm a bit sensitive here because those were hypotheses which just were thrown in the air and there was no experiment done. We were the first doing
it. Published in Nature, so I'm not sure.
Look at it.
It's thrown in the
air. Number one, we're not seeing much influence on the surface of the rock in the experiment. Number two, if you really go for the deep ocean, you'll have a trouble of making RNA because you don't have that dry state availability. And what you see here, what we try to do is checking hypotheses by experiments.
Therefore, I'm telling you, this is consolidating the hypothesis, because the hypothesis is still valid since years, and you are consolidating with your work.
I strongly disagree, but that's a longer debate.
Okay.
No, seriously, because people made hypotheses before the experiment, and that's always difficult to do.
Hypotheses have to be just proven, yeah, and this is
No,
exactly
you can only disprove hypotheses.
Disprove or proof. You have both ways.
Longer discussion also. I don't want to hitchhike this discussion here for now. No, you can only disprove hypotheses. I'm serious.
I wonder because I see that you have done almost all this research by focusing in the replication of RNA. But do you think that using similar system could work also for polymerizing peptides, or how do you see that? Do you think it's feasible, or because somehow, okay, you say that, okay, amino acids just are there and somehow make companion of the RNA, but the point is peptides has a lot of information too. Do you think this type of system could be used for studying the polymerization of amino acids and see how peptide gain in functions with polymerization?
I mean, people discussed and hypothesized without actually disproving with experiment enough that you get networks from even simpler molecules than RNA. And I just don't see how you get a reaction, an autocatalytic reaction network from peptides. That one peptide is making the polymerization of another peptide, right? Or how the information is maintained by it. People talk about formose reactions at pH 14, and those are not able to maintain information and can't do a autocatalytic networks. The idea is actually that only at the rather complex level of RNA, which needs a dry state, you can talk about autocatalytic networks. And the ligation chain reaction is one example of that, which directly leads to then the evolutionary pathway, which hopefully gives us the ribosome. So it is a bit a complex system, but I don't see how you can get that autocatalytic memory effect with more simple molecules. Happy to be disproven, right? But that's where we stand experimentally.
Okay, thanks.
Can I ask a question?
Sure, please.
So the question has to do with the dry cycle. And I was wondering, when you have a submarine hydrothermal system, the effluent that comes out is a mixture of liquid and gas. And if you have a chimney with a porous structure, it is not inconceivable that you would accumulate gas bubbles also in your pores and even growing gas bubbles. Wouldn't that work for you?
I mean, we did that high pressure experiment. If you go 200 bar and you have air bubbles in your solution, they immediately dissolve in the solution in water. You don't get air bubbles at high pressure. They get dissolved in the water.
Okay.
Look, I never say no, but you have to go then deep into the rock itself and where the water activity really drops, and perhaps there's something needs to be shown, but yeah.
No, I see your point.
Yeah,
it makes sense.
Yeah.
I
No,
guess...
that doesn't mean that hydrothermal on shallow, on Iceland, you get hydrothermal vents right at the surface. Of course, you'll have gas bubbles in those.
Okay, cool. Thank you.
You're welcome.
I have a very similar question to the previous one regarding peptide autocatalytic reaction. I understand that, this peptides could not hold the information, and therefore they cannot be specific. But whether it's really necessary that this network or these molecules are really specific. Maybe at early stages, these catalysts are unspecific. So they favor the formation of a lot of similar organic molecules, and specificity evolve during natural selection. And then also DNA appear at the stage when specific increase.
Look, we are looking a lot into, as we see that single amino acids help the polymerization of those 6-7 mers.
Yes.
We're looking into shorter peptides, whether they can help the templated ligation. And that would, make the cooperation between amino acids, peptides, and RNA put to the next level. So far, we have not seen something, but hopefully we get that. Those would be then a nice way to see how it collaborates. But the information side will be still on the RNA, and then you have to, but it would be nice to see that, because it could give you already very, very simple proto translation to then encode those peptides, 4 mers, 8 mers, from RNA, already a big advantage of your system, and therefore guide you towards ribosome and translation early on. I think that's necessary, because otherwise, the ribosome would have no advantage at the very beginning. I hope and think that there should be something.
Okay, thank you.
Many possibilities to probe. Yeah.
Okay, clear. Thank you.